Good afternoon. My name is Taran and I am a sophomore biology major.
My name is James Hermens, and I'm a junior biology major.
My name is Daniella Segall, and I'm an exercise science pre-physical therapy major. Before I begin, I would like to thank the judges and organizers of the symposium for the privilege of the podium today. We will be presenting our project to you, the 12 Phases of Autism Spectrum Disorder, otherwise known as ASD. We report no conflicts of interest. As you can see on this diagram over here, it looks like a complete mess and a complete array of different things. Well, this is what ASD is. ASD is a hodgepodge of symptoms that can range from things such as not responding to your name all the way to cognitive disabilities and seizures. Two symptoms on very different ends of the spectrum. And because of this, we cannot treat all ASD patients the same, as they fall differently on the spectrum, and therefore, no two patients will present exactly alike. This is where the classifications for ASD in DSM-4 and DSM-5 differ. DSM-4 required deficits across 3 domains, these being social, communication, and restricted interest. DSM-5 took these factors and reduced it down to 2 categories, being social communication and repetitive behaviors. As you can see on this diagram over here, in DSM-4, ASD fell under one big umbrella called persuasive developmental disorders. DSM-5 removed all the specificity and created one big umbrella called ASD. Due to this lack of specificity, as you can see here in the graph, there was an exponential increase in ASD diagnostic rates in 2013 when DSM-5 was published. In 2013, 1 in every 88 children had been diagnosed with ASD. Fast forward 13 years later to now, 1 in every 31 children are diagnosed with ASD. This is going from a 1.25% diagnostic rate to 3.2% in present day. This is almost double. So, as I previously said, there was an exponential increase in ASD diagnostic rates. This is because due to a lack of specificity. We mentioned this term a lot in our presentation, and you might be wondering to yourself, what does this mean? As you can see on the slide behind me, we have 3 patients all having 3 different symptoms. Patient 1 can engage in daily activities, struggle with social communications, as well as planning and organization. Patient 2 requires support 24/7 for daily tasks, is 10 years old and still in diapers, and has severe communication impairments. Patient 3 is socially isolated, struggles with repetitive and obsessive behaviors, and is still able to have some form of pronounced communication. All 3 of these patients are diagnosed with ASD, yet all present differently. They are all treated with a similar combination of treatment, which we'll get to later in the presentation. But before we do that, we need to ask ourselves, do all 3 of these patients require the same thing? This brings us to present-day procedure. It typically follows a 4-step diagnostic procedure. The first is to identify whether they have persistent social communication and interaction deficits. So these are things such as failure to initiate or respond in social interactions, as well as deficits in nonverbal communicative behaviors. Step 2, do they have any repetitive behaviors or patterns? This is things such as repetitive motor movements or speech, insistence on sameness, hypo or hyper reactivity to sensory stimuli, as well as highly fixated interests. Step 3, symptoms must be present in early developmental period. And step 4, symptoms cause clinically significant impairment. However, we tried to look at shifting the diagnostic procedure away from behavior-based observations. If we can look at the genotype that correlates with the ASD phenotype, this could allow for biomarker-driven classification. This takes us to our treatment in present day for ASD. There is no cure, so we tend to treat the symptoms. Ideally, treatment is carried out between 18 months to 3 years old. However, if we were able to do genetic testing at an earlier stage and correlate the genotype with the ASD phenotype, this can help us get better results in treatment. Like I said previously, treatment is carried out in a combination of things. This includes behavioral and communication therapy, educational therapy, and pharmaceutical therapy. Now that you know the current options that are provided today, we are going to explain what we did to try reshape as well as refocus how treatment and diagnostic procedures are carried out for ASD in a true translational medicine approach. This leads to our specific aims we had for our project. Number 1 was to evaluate the relationship between gene expression and distinct phenotypes of autism using a genotypic atlas. Number 2, to identify distinct subtypes of autism based on which symptoms tend to occur together in people with specific gene variants. And step 3, to analyze the clinical impact of the subtypes of autism. I'm now going to hand over to Taran to explain a little bit more.
As my colleague Daniella mentioned, the purpose of our research was to correlate the ASD genotype to the phenotype. The next 2 slides provide a framework for our research. It is a representation of how we were able to separate ASD into 12 distinct categories. In the primary stages of our research, we harvested all the genes associated with autism from SFARI, also known as the Simons Foundation Autism Research Initiative. SFARI works with families and affected individuals to provide a better molecular basis and understanding of autism. This provided a basis for our research. Each gene obtained from the database includes a name, a symbol, an ID, a chromosome location, and an evidence score, which I'll explain later. Following the collection of the genes from the database, we purchased papers from PubMed using NCBI Entrez API, an interface that provides access to PubMed papers. We set parameters to remove all non-human studies. Genes with 5+ supporting papers were selected for analysis, yielding 241 genes with sufficient phenotypic data for robust clustering. We then placed all the papers we'd obtained into an AI called Claude AI. The AI analyzed the papers and organized them according to similarities and phenotypes aligned with the DSM-5. Each phenotype that was in agreement with the DSM-5 was assigned a status: present, absent, or not reported. Then a confidence score was applied between 0 and 1, indicating how reliable the paper's information was. Along with each gene, a snippet of evidence was included, and this can be seen here at the bottom of this slide. This was extracted from the SLC6A4 gene. Following these initial steps of collecting and analyzing data from the SFARI database, the AI began to sort the 1,053 papers from the aforementioned 241 genes, and hence we developed the 12 faces of autism. The QR code at the bottom right of the screen can take you to the genotypic atlas that we created. The methods of data harvesting can also be found on this website. Currently, we are progressing with manually checking each gene that the AI placed into our database. The creation of the 12 faces of autism could change how ASD is diagnosed. However, it wasn't just luck or coincidence that allowed us to discover these categories. In order to sort the 1,000+ papers that we obtained, we used 2 forms of statistical modeling. The first, Bayesian-Gaussian mixture modeling, and the second, Dirichlet process prior. Let's start simple with Bayesian modeling. Imagine you have a huge bucket of Lego bricks, and you're trying to sort them based on color and shape, but you don't know what colors and shapes you have. If this was a computer simulation, Gaussian modeling would allow you to assign each brick a home. I'm going to break this down further into 2 parts. What is Bayesian modeling, and then what is mixture modeling? I'll then describe how they helped assign the 12 faces of autism. Bayesian-Gaussian modeling describes a statistical approach that uses Bayes' theorem to update the probability of a hypothesis as more information becomes available. This model combines observed knowledge with data to produce a probabilistic model explicitly accounting for uncertainty in predictions. The Gaussian aspect of this model uses distributions, also known as bell curves, to predict a range of possible outcomes rather than a finite result. Next, we look at mixture models. This is a form of probabilistic model that is used to represent the presence of subpopulations amongst a larger population. However, it does not require that the observed data set identify the subgroups of the population. This form of modeling also demonstrates the likelihood of a data point belonging to more than one group, identifying data points on proximity to multiple different clusters. Now, putting this all together, Bayesian-Gaussian mixture modeling is used to fit a number of unknown parameters. For example, let's assume the AI is reading a paper on the SLC6A4 gene. As it reads the paper, it slowly gathers information about the phenotypic expression of the gene. As information is continually uncovered, the AI adapts and adds the gene to the most appropriate cluster, whether it is a new cluster or a currently existing cluster. If the gene presents more than one phenotype, the AI aims to place the gene in a subgroup that best fits, but also in proximity to a subgroup that represents the other phenotypes. Now we look at Dirichlet process prior. This is a common method of Bayesian statistics where clustering is suspected amongst random variables, but we do not know how many clusters there are, nor which random variable belongs to which cluster. Using this form of modeling, the AI is able to assign genes to clusters as it reads information or even create new clusters. This model uses a Bayes distribution, which suggests the tendency of a cluster, but the Dirichlet process allows the cluster to develop elsewhere. Essentially, this is a form of analysis that encourages the growth of developed clusters while simultaneously allowing for the creation of new clusters. Using these probabilistic models, the AI was able to create 12 clusters, which we coined the 12 faces of autism spectrum disorder. But this was not the end of the road for the AI. Now that we developed a framework of subgroups, the data and classifications had to be validated. We were quite surprised to see 12 distinct subgroups that at first we didn't believe the results. We were worried about overfitting, which is where the model does too good of a job, it no longer reflects reality. This was a key part of the project, and we had to make sure we got it right. As a result, we used two different approaches to validate our data. The first approach we used was standard machine learning classification model. How this works is that it trains an algorithm on a subset of data to construct a model. It then uses the other half of the data to determine whether the model it generated was accurate. It repeats this many, many times until it is able to construct a high-quality model that explains the variance in the majority of the data. The specific machine learning model we used was called a random forest tree, and it specifically explained 86% of the variance and arrived at the same conclusion as Bayesian-Gaussian mixture modeling and the Dirichlet process prior. What was most impressive about this model is that it is a completely separate way to analyze our data, but also produce the exact same results. The other process we used wasn't quite as strong and was also prone to overfitting. This is called latent class analysis. This is basically a fancy classification model where it repeatedly probes the dataset looking for differences. It does so until it has managed to find all the differences and split the data into distinct clusters and groups. Despite its flaws, this model produced the exact same results as the previously used models. It wasn't as meaningful as a test as the random forest model, but it was still an important additional check. Now, you might think this graph looks a little ridiculous. It's hard to read and honestly an overload of information. This is a representation of all the phenotypes we discovered through our research. Now imagine entering this data manually. It would take a very long time. The AI assisted with classifying the 12 phenotypes of autism, which my colleague James will explore further.
Through thousands of papers and thousands of patients, I give you the 12 faces of ASD. We have 6 minor clusters and 6 major clusters. The 6 major clusters being dyspraxia, deficit in motor planning, GDD or global developmental disorder, pure intellectual disability, language impairment, seizures, full ASD syndrome, as well as behavioral and anxiety core. As said, we looked at 1,053 papers, and our machine learning was said to be 86% correct, which we did find statistically significant. This is a graph that illustrates our gene-to-trait distribution. Intellectual disability had by far our highest number of genes associated with the phenotype. Major clusters had over 50 genes associated, while minor clusters had 7 to 8 genes associated with them. This is a picture coming straight from the website that we created. Right now, we've ataxia clicked on, which is a problem with coordinating movements. On the left is the list of genes that are associated with ataxia. So not only do we have broad faces of ASD, but we also couple those with syndromes or phenotypes that show up within that cluster. Finally, that brings us to the patients that we saw in the beginning of the slideshow. We not only see the symptoms that they present with in the physical world, but we know what happens at the molecular level, and we have answers for the symptoms that impact these children's daily lives. The impact that this has largely affects the diagnosis and treatment of ASD, as there's multiple different subtypes, each of which completely changes treatment and diagnosis. Different subtypes have different neurotransmitter profiles as well as genetic drivers. We have true translational medicine here coming from the lab bench with clinical applications. Pharmaceutical treatment, as of the modern era right now, largely relies on treating broad symptoms, such as prescribing a stimulant to a child with hyperactivity. With this research, we also have a possibility to conduct early intervention. As previously stated, therapy is most effective when carried out between the ages of 18 months to 3 years old. Our research gives physicians something to look out for in terms of early risk management. Each gene that is associated with a phenotype of ASD has a signaling cascade pathway associated with it, as seen in the figure. And these are things that we could already have medicine for as a biomedical intervention for ASD symptoms specifically. So, in conclusion, separating ASD into different phenotypes and categories has the potential to reshape clinical care for our affected populations. Results from genetic testing allow physicians to not only look out for the spectrum of ASD, but specific symptoms and phenotypes. This work allows for specificity of either pharmaceutical treatment or therapy carried out in children's lives. For future directions, we are to conduct a comprehensive assessment by hand of every paper listed under the database to confirm the phenotypic data, as well as isolate a gene that is strongly associated with a phenotype, evaluate its signaling cascade, and see if we have any modern-day medicines that can affect the signaling pathway. We would like to say thank you to Dr. Desai, Dean of the Library, Katie Krauscher, judges and organizers of the academic symposium, those at the Simmons Foundation, as well as our human investigators, Jackson Jones, Angelica Rose, and Chantal Tissienga. Thank you.